Kyle and Alex, you all are impressive draws. I would have put the over under on this attendance probably south of 50, and we are sitting here at 78. So, um, really impressive turnout to everyone on the phone, on the webinar, joining so soon after the holiday. So, Happy New Year to everyone. We are really excited about this uh, two topic webinar we've recruited today. They go from Lake Superior down to Lake Ontario with the common theme of Cisco telemetry and some of the challenges associated with tagging Cisco, but then some of the really valuable insights that we can learn once those methodological challenges are overcome. So leading off today is going to be Kyle Stratton. And I'll just give a brief introduction of both Kyle and Alex, and then they will give back-to-back webinars, and then we will hold all questions until the end. I think Jake also will post the registration for our February webinar, which we're also really excited about. Hopefully we'll have another great turnout for that. And he'll sort of share that poster at the end of these talks as well. So first, Kyle is going to be talking about telemetry on the North Shore of Lake Superior. Kyle got his undergraduate degree at Lakehead University, stayed at Lakehead for his master's degree, working with Mike Rennie to study what could be underlying the decline in steelhead in Black Bay, Lake Superior, using life history theory and isotopes. He became a fisheries technician in 2020 for the Upper Great Lakes Management Unit of the Ontario Ministry of Natural Resources. He then became a biologist intern in 2021 at that same office. And since 2022, he has been a project biologist for that management unit. So we'll hear first from Kyle, and Alex, who we'll hear from second, got his bachelor's in biology from Hobart College, and then went to Purdue University to get his master's degree in fisheries science, working with Thomas Hook on a project that sought to restore spawning habitat for lithophilic spawners in Saginaw Bay. 2019 to 2023, he was a fish biologist with Fish and Wildlife Service, the Lower Great Lakes FWICO, the Native Species Program. And in 2023, we were able to recruit him to be a new biologist at USGS, Tunison Lake Ontario Biological Station, which is in Cortland, New York. And Alex's focus there also remains on native fish restoration. So with that, I will turn the floor over to you, Kyle. Each of these talks is about 15 or 20 minutes. And again, please stay on for questions and comments after these talks. Thanks, everybody.
Thanks for the introduction, Bo. So, like you said, my name is Kyle Stratton. I'm a biologist for the Ministry of Natural Resources in Thunder Bay. And today I'll be presenting some of the work that we've been conducting on Cisco acoustic telemetry. So my goal for this talk is really to provide prospective researchers with some potential ideas or considerations if you're considering doing telemetry on Cisco in Lake Superior. As such, most of what I'm going to be presenting here today is going to be focused on some of the methods for how we caught Cisco, what worked and what didn't work. But why Cisco in the first place? Why are we conducting telemetry on Cisco anyway? So Cisco are a very important prey species in Lake Superior. So to sort of emphasize this, the role that they play in the overall fish community, we've recently experienced the largest Cisco recruitment event that we've ever detected in Lake Superior. Just happened a few years ago. And as a result of this event, we've seen some pretty drastic shifts in some of the sizes of some of the fish that we're seeing show up around the lake. One example being the unprecedented coho and Chinook salmon sizes. We had Chinook salmon this year that were pushing 40 pounds in Lake Superior, which is, I would say, a 3 or a 4-fold increase over the average size Chinook that we typically produce up here. And I think Minnesota broke their coho state record 3 or 4 times in the last year. Just as an example of this here, this image on the left is about 7 or 8 Cisco that were found in one of the stomachs of Chinook salmon that we captured through one of our derby monitoring programs. So they play a very large role in the prey base of the lake. Additionally, cisco represent a very important aspect in the commercial fishery, as they're the second highest valued species in the commercial fishery within the Ontario waters of Lake Superior, second only to Lake Whitefish. So the commercial cisco fishery is a roe fishery. So these fish are targeted leading up to and during the spawn, and the roe is shipped over to Scandinavian countries. So we started our Cisco telemetry project in March of 2023. During the first year of this project, our focus was largely proof of concept. So this included determining how to catch, tag, and release Cisco successfully. And our fish tagging objective this first year was to tag and release a total of 50 Cisco. So we wanted to tag 25 Cisco in Thunder Bay and 25 Cisco in Black Bay. And during the second year of this project, so this would have been 2024, our goal was to tag an additional 25 Cisco in Nipigon Bay. So the overarching goals of this Cisco telemetry project are to address whether the Black Bay and Thunder Bay Cisco populations should be managed as a single stock or as two discrete stocks, as well as identifying spawning time, site fidelity, seasonal movements within and between bays. And lastly, to determine how susceptible the Cisco stocks are to commercial fishing. I'd just also like to mention that before we started tagging any Cisco, we did already have an existing acoustic receiver array in place. But as I mentioned, the focus of the talk that I'll be giving here today will largely be focused on the methodologies we use to catch, tag, and release the Cisco. So we captured our Cisco on Superior during the late winter, so February and March. So our reasons for targeting Cisco during the late winter were, one, we wanted to catch them after they had spawned. So Cisco spawn in the late fall. The females are pretty fecund, so their body cavity is fully occupied by eggs. This would make it very difficult to insert a transmitter into the body cavity. And I imagine that it would also be extra hard on the fish. Additionally, spawning times are already a fairly difficult time for fish. So having fish that are fully healthy to spawn as opposed to one that's recovering from surgery, it just made sense to help improve survival. We also wanted the water to be fairly cold for healing purposes. And then lastly, we wanted to be able to keep the Cisco overnight after surgery to assess the recovery prior to release. And keeping them overnight was logistically going to be much easier for us to do during the winter in sort of a controlled environment on frozen Lake Superior than it would have been during the open water season. And as it turns out, and I'll touch on this a lot more later, keeping them overnight had some very significant implications on their survival. So we tried two different methods of capturing Cisco through the ice. So the first method we used was using a canned gill net right under the ice, as I've tried to illustrate here. So we would drill several holes side by side, and we would use a jigger board to run a line about the length of the net under the ice. And then we would drill a bunch more holes, get the jigger board up, attach the line to the net, and then pull the net under the ice. The net itself had multiple floats on it, so it would float up against the bottom of the ice. And we also added more floats to the top of the net just to help with buoyancy. The net was 1.8 meters tall, so about 6 feet tall. And we were typically fishing in about 10 to 30 meters of water, but again, right up along the bottom of the ice. Now, as most of you probably know, gillnets are very hard on Cisco. So to combat this, we would let the nets fish for no longer than 3 hours before lifting them. And then to remove the Cisco from the net, we use scissors to cut the mesh. So once we had Cisco in the net, we'd take them out, cut the mesh, and then we'd place them in a cooler that was full of water. And then once the entire net was lifted, we'd shuttle all of the Cisco in that cooler to our surgery or fast recovery tent, I guess, if you want to call it that. Now, the size of the mesh that we used was 3¼ inch, And the reason for that is it's the same mesh size used by the commercial fishermen that are targeting Cisco in Thunder Bay and Black Bay. Now, the second method that we tried to use to capture Cisco was angling. Um, and I'm not really going to touch on that too much, but ultimately we were pretty unsuccessful at consistently catching Cisco via angling. So the gill net really proved to be the better way to capture them. So these are just some images from the field operation. So the image here on the left, this is what our jigger board looks like under the ice. So again, this is how we would run the line under the ice to be able to set the net. The middle image is of myself and a coworker lifting one of the gill nets up in Black Bay. So you can see all the holes that we had to punch to be able to get the net in and out. And the cooler here. So the cooler was really important because the first Cisco we caught, we just put them in a bucket and then we strapped them on the back of the snow machine and then we tried to drive them over to our our tent with the holding pen in it, but there was no lid on the bucket. So when we hit a bump, we shot the Cisco right out of the bucket. So any sort of tote with a lid on it, as long as it's filled with water, the Cisco seem okay with it. The sloshing around doesn't really seem to negatively influence them too much. And then the image here on the right is looking down through the ice, and you can see the floats here on the net, and then this gold colored thing is actually a Cisco that we caught in our net. So once we have the Cisco in the cooler out of the net, we bring them to our surgery recovery tent, which I've shown here on the left. So all it is a large pop-up ice fishing tent. Inside the tent, we drilled two large holes about 1 meter in diameter, and we put in mesh nets to hold the Cisco before and after surgery, which you can kind of see in the image here on the right. So this is what our holding net or holding pen would look like. It's about 1 meter in diameter and it stands about 6 feet tall. So the Cisco's, once we caught them, we would put them in one of these nets. We would do surgeries on them the same day, and then they would go into the other holding net. So the day after we did the surgery, we would assess the Cisco's for the ones that lived, and if they lived and they looked like they were okay, we would release them. If they died in the holding pen, we would just take the transmitter out and use it on a different fish. The one thing that I'll mention is that our release location was very close to a deployed receiver. And the reason for this is we wanted to be able to see if the Cisco swam away once we released them or if they just went right down to the bottom where we would theoretically continually detect them on that one receiver. So we could assess whether or not they lived. So for transmitters, we used V9 predation tags. So although the Cisco that we were catching were all fairly large, I would say between 40 and 50 centimeters, we decided to use V9 predation tags as we thought that there were quite a few predators that were capable of eating even a very large Cisco. So a really good example of this is this lake trout here on the left. So this lake trout was actually captured in one of our canned gill nets, and he was about a foot behind a Cisco in the gill net. So he was obviously trying to eat that Cisco. We also caught a few other trout in that net as well. So ultimately, we really just wanted to be sure that the fish that we were tracking were actually Cisco and not something that ate a Cisco. The image on the right is of a couple of pike that were captured in Black Bay in a different assessment, but these pike are surely large enough to eat any of the Cisco that I've ever seen. So for surgery, well before we even got out on the ice and started trying to catch Cisco, we were practicing surgeries on dead Cisco that we caught in our other field programs. So although doing surgery on a dead fish is quite different from a live one, in my opinion, it is important to go through the motions of practicing to do the surgeries just so that you get the repetitions in and you feel more comfortable once you're actually doing surgery on a live fish. For a surgery trough, we built it to be customizable depending on the size of the Cisco, because there was some fluctuation in the sizes of them. We also used a water pump to ensure that there was always water going over the fish's gills during surgery. And then for immobilization, we used a TENS 7000 unit, which we purchased on Amazon, and it worked very well. Now, if anybody's seen the TENS pads, there are sticky adhesive pads on the unit. But we covered them with tinfoil to prevent them from sticking to the fish, and that seemed to work quite well. For our other telemetry projects, we use the electro immobilization gloves, but for the Cisco, being a smaller fish, not having a pair of hands actually touching the fish made the surgeries go much better, in my opinion. So let's take a look at the success rates or lack thereof from 2023. So the first year that we were doing this project. So the number of at-large and eaten Ciscoes here on the far right of these plots are as of October 2024, or roughly 1.5 years after initial release. So if you look at the top image and you look at the bottom image, one takeaway here is that we improved our ratio from captured Cisco to at-large Cisco pretty significantly. And initially, I attributed this increase in Cisco survival to us simply having more experience as we started in Thunder Bay and then we moved to Black Bay. But having a little bit more information now, I think there was something else here at play, and I'll touch on that in a little bit. Now, for Cisco that were eaten, this is kind of interesting that 82% of them were eaten less than 1 month after release. So to me, this suggests that the Cisco that we released were likely injured or recovering for roughly 1 month after release, which the predators really capitalized on. And the other takeaway here is this really shows the importance of using predation tags on Cisco and Lake Superior as a really high number of our Cisco were predated upon. Much greater percentage of them than we had initially thought before we started actually putting tags in Cisco. So when I'm saying at large and died post release, how did I determine that? So again, we released Cisco very close to an existing receiver. And knowing something about Cisco movement before this, with them being a pelagic fish, I think we would assume that they would be moving around quite a bit and getting picked up on likely multiple receivers. So for Cisco that are considered at large, as you can see here, they were detected on multiple receivers, whereas this Cisco, I'm considering it dead, and it was only been continuously detected on the one receiver directly beneath where we release them. Although this method might not be um, the best way of assessing if a fish lived or died for other species for telemetry projects, I think that it makes sense for Cisco in this case. Again, just because uh, they would move around a lot more than, say, something like a yellow perch, I think. So, does the net pen increase post-surgery survival? Short answer is no. So, 30-day survival rates for Cisco that spent one night in the net pen after surgery was 27%. Compare that to the 30-day survival for Cisco that were released immediately after surgery, it was 65%. So if you think back to how our survival improved greatly between Thunder Bay and Black Bay, in Thunder Bay, all of the Cisco we kept overnight prior to release. Whereas in Black Bay, most of the Cisco we released the same day as the surgery, either because we captured them on a Friday and we didn't want to keep them in the net pen over the weekend, or we captured them on the last day of safe ice. So we just released them the same day as the surgery. Now for Nipigon Bay in 2024, we saw the same thing that individuals that were kept overnight exhibited far lower survival than those that were released on the same day. And we'll see if this video plays here. So this is just a quick video of the release of one of the Cisco. Okay, so let's take a look at some really quick preliminary movement results. So this map shows the number of detections at each receiver. So Cisco that were tagged in Thunder Bay on the left here in red, Cisco that were tagged in Black Bay are in the middle in yellow, and Cisco that are tagged in Nipigon Bay are in the top right here in green. So one of the main movement results so far is that none of the Cisco that we tagged in Thunder Bay have entered Black Bay. Conversely, some of the Cisco that we've tagged in Black Bay have entered into Thunder Bay, but they didn't spend much time there. And then Cisco that we've tagged in Nipigon Bay really haven't left Nipigon Bay much, and if they did, they didn't go far. Granted, this is only about 6 months of data for Nipigon Bay fish, so this still could change. There have been a couple of interesting fish as well. So one of our Cisco that was eaten in the Bay's End area of Thunder Bay ended up down towards Grand Portage in Minnesota. I don't know what ate it, probably a lake trout, I guess, but whatever it was, ate it and then took off for about 60 kilometers. And then another Cisco that we tagged in Black Bay ended up swimming all the way to the mouth of the St. Louis River and then coming back to Black Bay. So there has been some pretty large scale movements by some of the fish, which I think we would expect. So main results thus far. So predation rates are really high. So overall, our predation rates are about 36%. So this really highlights the importance of using predation tags if you want to tag Cisco in Lake Superior. Secondly, using the net pen to keep the Cisco overnight was a complete bust, as all it did was really lower their chance of survival after release. Maybe if we had a bigger holding pen, it would help negate that. But then we start running into other logistical challenges. So this year, when we go to tag, hopefully tag 25 Cisco south of Thunder Bay towards the Ontario-Minnesota border, we won't be having the Cisco overnight in the net pen. We're just going to do surgeries and release them immediately, and hopefully we'll see a bump up in our survival rates. So lastly, I just want to show what our current array looks like. So it spans all the way from the Ontario-Minnesota border here in the bottom left corner all the way to Terrace Bay on the right side of the map. We don't have receivers going out further past this line because this line is about the 100-meter contour depth line. And because we don't use AR receivers, we don't go beyond 100 meters because then it just gets too difficult to get our receivers back. There's my email. If anybody has any questions, feel free to send me an email. Otherwise, that concludes my portion of today's seminar. Thank you.
Alex, oh, there you go. Okay.
Yep. You want me to just jump right in here?
Jump right in. Yeah. Great job, Kyle. That was awesome. I got questions. I'm sure others do too.
How's that showing up for you?
Flip that. Do your switch. There you go. Swap. There you go. Perfect. You got it.
Okay. All right. Thanks very much, Bo and the Fish Commission for the invitation to give a quick presentation today. I'm just going to switch gears a little bit to Lake Ontario and how we're using acoustic telemetry to answer some of the questions around habitat use and some spawning behaviors for Cisco. So there's a bunch of people that have worked on this. We've published it now, and so it's been a few years since the study is concluded. But I just want to acknowledge that what I'm going to present today came from, it was led by the U.S. Fish and Wildlife Service, so that was through Dmitry Gorski and Ty Biesinger here. I didn't join the team until a little bit later on in my previous life as a fish and wildlife biologist. So the props go to these two for the setup and design of this study. We also had help from the USGS, New York State DEC, and then, of course, GLaDOS for data management for this project, and it was funded to the GLRI. So that just give a really quick, dirty overview of Cisco and Lake Ontario, just to get everyone caught up if you're not already there. We're going to be talking about Coregonus ardii today. So this is our native pelagic planktivore in Lake Ontario. Similar to the other Great Lakes, Ontario's populations are reduced from a handful of things like habitat degradation, overfishing, invasive species. But fortunate enough for us in Ontario, we didn't have to overcome the same types of local extirpations like Lake Erie here with that famous picture of the Earl Bess. So in current Lake Ontario, we have remnant populations. They're mostly delegated to the eastern basins of Lake Ontario. And we do have some recruitment, but it's pretty sporadic. So if we look at some of Brian Weidl's bottom trawl surveys from USGS, DC, and OMNRF, we can look at the catch per unit effort of adult Cisco throughout the sample years of the 80s until current. And over the last 20 years or so, really the only recruitment that we're seeing is coming from the eastern basin. So that bottom panel there. Because of this and all of the interest in restoring biodiversity to our forage basin, Lake Ontario, Cisco has kind of landed on that list as one of those species that restoration is a management objective for in our lake. The two more important, we'll call them important populations, spawning populations in Lake Ontario are located, one in the US and one in Canada. They're both highlighted here in the red circles. So Chameau Bay is the bay on the US side, and Bay of Quinte is on the North Shore on the further west side there. In Taylor Brown's master's dissertation, her and others put together some really nice data on larval densities spread out across Lake Ontario. And what they found was that Chameau Bay had some of the highest larval densities compared to other parts of the lake, especially Bay of Quinte as well. Though there was still some production coming from other parts of the lake, the largest densities were still Chameau Bay. The other important aspect of this population was that Chameau Bay, for a brief period of time, for about 10 years, USGS and others had some experimental stocking in the south shore of Lake Ontario in US waters, and the Chameau Bay stock was used to provide animals for that experimentation. So we know quite a bit about the Chameau Bay population, but there were still some questions that remain that we could answer using tools like acoustic telemetry. And some of those were seasonal migration pathways where these Chameau Bay fish when they're not in Chameau Bay. The spawn timing and location. We know that they spawn within the bay itself, but we don't know about those smaller micro habitats where they might be using some rather than others. And then finally, what does spawning site fidelity look like? Is there mixing of populations between Chameau Bay and Bay of Quinte, or some of those kind of populations just holding on in the southern embayments as well? So we thought that acoustic telemetry would be the appropriate tool to answer these questions. And by answering those questions, around spatial distributions, we might be able to help the restoration efforts by the managers. So be able to directly hand them some information that might be useful with their management decisions. There were two study objectives for the research I'm going to present today. The first is kind of more general, is to describe Cisco's seasonal habitat utilization related to depth and temperature, and then also identifying some of those regional migrations. The second was a little more specific, to identify Cisco spawning sites within the bay itself, and then to determine the spawning site fidelity of this population as well. To go into methods really quick, Kyle did a really nice rundown of acoustic telemetry, so I don't really have to get too into the weeds here. We use some of the same methods that Kyle was talking about a little while ago, and then we had some differences in what we did. But we were able to track the seasonal movements from 2016 to 2021. We had originally tagged 76 reproductively mature Cisco during the spawning season. So from the figures I showed previously, it's pretty hard for us to catch Cisco unless we go at spawning times when they're concentrated. And so this led us to the decision to, one, we wanted to make sure we were tagging Shimo Bay fish or fish that were there during spawning time. And B, that's the only time we could really catch these fish. So that's why we decided to tag the fish during November and December when they were readily available. So we used a mixture of different tag types, some pressure, some temperature, some had pressure and temperature, which allowed us to get that bathythermal niche data, which I'll talk about in a second. The fish were tagged right at spawn, so it was the last week of November, first week of December. We had a fine scale array within Shimo Bay itself, and it moved around a little bit from year to year. And then we used the GLaDOS network of receivers out in the main basin so that we could get those larger movements and outside of Shimo Bay movements as well. And then importantly, when we're doing our data analysis, we use centers of activity, which uses trilateration of telemetry data. Basically, it means if you're detected by more than 3 receivers or 3 or more receivers, we can use math to give us an estimate of more fine scale positioning of that fish. And this comes into play when we're doing fine scale telemetry within Shimo Bay itself to try to locate those smaller habitats where the fish might be utilizing for spawning. The other thing that we wanted to do was ground truth all our telemetry with actual gamete collections, right? The fish are in an area we think they should be spawning during the time, but we need the actual hands-on physical evidence that they were spawning in those areas. So Brian Weidel and others at USGS for the last 10 years or so have been doing a really great job of collecting gametes in Shimo Bay itself. So we were able to pair our data with egg collections from Brian's team in 2019. So we had set 105 traps throughout the spawning periods. That was end of November to midway through December. Those traps were only pulled one time just to get a bulk number of egg deposition through the spawning period. And we use shoals and suspected spawning areas that Matt Brevet had identified in his master's, and Ellen George, now Ellen Karboski, had identified during her PhD as well. So those were kind of our starting points of where we should go to look for Cisco eggs. This figure gives you an idea of our study location. So a little bit of warning here, this array changes every year. So this is just one representation or 1 year's situation for the array. This isn't necessarily every year. But each one of those points is a different acoustic receiver, and we have them grouped into zones that made sense for our data analysis. So a main lake zone, which is the lighter blue, this furthest west zone, which was the Rochester red, Bay of Quinte, which was orange, the Black River Channel, which is kind of a staging area before fish get in, start moving into Shimo Bay. That's in yellow. And then our Shimo Bay fine scale telemetry array, which again moved from year to year, but in general, we had about 27 receivers in this embayment. And the yellow stars there indicate the locations where Cisco were trap netted, tagged, and then released in those same areas. So we did not use gill nets. In this case, we used trap nets because we had been using this collection technique for those broodstock collections that I spoke of earlier for the USGS experimental stockings. Okay, so we'll just jump right into some results. This is what people like to see first, right? Animations of fish moving around. I'm going to start with just a single year. It's a lot easier to view this way. Before I let this thing run out and you guys get to see the fun movements of the fish, I just want to mention that though we tagged 76 Cisco throughout the 3 years of tagging, we only had survival of 23. So Kyle touched on this a bit before. Fish during spawn are stressed out. It's harder to get high survival. We tagged both males and females in this situation, and the females didn't make it. So we didn't have a single female live past 3 months to get any useful detections from. So everything you're going to see from this point on is based off of males and not females, is a little bit of a caveat here. Okay, so we're going to start January 1st, 2020. This is after the spawn. So some of the fish within the population stay within Shimo Bay throughout the wintertime, but some move back out in the wintertime to their feeding grounds. As we get later into the spring, water temperatures start to warm in Shimo Bay, kind of above the threshold for Cisco. They start to move out of the abayment by June. They're all out and in their foraging habitats for the summer. So as we move closer into the fall, when water temperatures start to become more isothermic, you're going to see fish start to migrate back from their foraging areas to the Black River Channel, which is that staging area, and then into Shimo Bay during the spawn. And then the cycle repeats as we get closer to January. So let me put my pointer on here. There's a couple of interesting things just right off the bat before we did any real analysis. We saw that there was groups of fish that that would have different migration patterns outside of the spawning season. So namely these ones down here in Rochester, and then there were a few fish up in the Bay of Quinte as well that had annual returns to areas that were not Shimo Bay outside of the spawning season. And so we'll talk about this in a little bit, but this might indicate something similar to foraging site fidelity as well. To go back to our objectives for our study, the first was kind of this broad scale. Let's get an idea of what the habitat utilization is. So we have a biplot here of temperature and depth from our tagged Cisco with these Bayesian ellipses around the averages. And so kind of the take home from this plot is that kind of not a surprise. In the summertime, these fish move offshores. They have availability of different temperature waters, right? They're a pelagic planktivore, so they're hanging out near the thermocline, above or below it, depending on where the food is at that time. So you see this larger ellipse during the summer. As we get into more isothermic conditions, the fish move near shore for spawning in the wintertime, and so their bathythermal niche is more constricted in the winter. The second part of our first objective was kind of define seasonal migrations, and we chose to do this in two different windows. So the non-spawning period, so this is anywhere outside of November, December, and then the spawning period, which was inside November and December. And what we saw with this node analysis here is the direction of movement. So that's which way the arrow is pointing, gives you an idea of which way the migration was. And then the thickness of that connective line between the two nodes tells you how many of our fish were making those movements, kind of an indication of importance of that migration pathway from our sample population. And so during the non-spawn season, you can see there's quite a bit of movement. They're moving some fish across the lake to Rochester area or up into Bay of Quinte. But when we get into the spawning season, all migration paths point back to Shimo Bay. And so this is telling us that we're not seeing any straying from the limited sample size that we had of just male fish. These fish were 100% returning to Shimo Bay. Moving on to the second objective, this was looking at spawning dynamics, spawning behaviors, and kind of the location of the areas that these fish are using to spawn. This is just a simple heat map showing where we're getting larger detections or centers of activity. So the darker colored red is our areas that more fish are detected on, and moving colder where less fish are detected. So using this type of information, we can get some just pretty simple metrics here. So average time spent in Shimo Bay, the average peak spawn dates. We're defining peak spawn here as the highest proportion of our tagged fish in the bay at any given time. And so in this case, because our site fidelity was so high at 100%, this is the period at which 100% of the fish were in the Shimo Bay. So this isn't to say that the fish are spawning from November 2nd to December 10th. It's just that they're within the bay itself during that time, which we're defining as peak spawn. So with that correlates to water temperatures starting around 8.5 degrees Celsius and ending just below 4 degrees. When we looked at the habitat beneath these areas that had the highest concentrations of centers of activity, It was on shoals with an average depth of about 3 meters, and this is what that substrate looked like. So probably originally rocky cobble, but now infilled by things like drusented mussels and drusented mussel hash. Our egg collections matched our acoustic telemetry really well, which was nice. So here in this bubble plot, the larger the bubble, the more eggs that were collected in those areas. And any of those red crosses are where traps were, but did not collect eggs. So when we compare that to our fine scale telemetry here, we can see that there's a pretty nice overlap with the egg deposition and the areas that we had the highest detections. So we're pretty confident in saying that where our fish were detected is where they were likely spawning in these higher COA areas. Finally, this is the messiest and admittedly one of the hardest figures to understand, but I'll just walk you through it really quickly. It gives you an idea of where fish are at certain times of the year. So on the x-axis here, we have the full range and dates from our study from January 2017 to July 2021. On the y-axis, we have the percent of tagged fish. So these are our fish that are live swimming around. What percentage of them are in each of these different zones, which are highlighted by the colors here. And so the most important part of this figure to look at as far as site fidelity goes is during the spawning time. So this is 4 different spawning periods here from November into December where these hash marks. And what you can see is that the dark color blue, which represents Chameau Bay here, is filled in 100% in each of these 4 years, which indicates to us that these fish are returning to Chameau Bay annually during the spawning time and not visiting other known areas where Cisco may be spawning. All right, so just to wrap it up here and kind of go back over some of our discussion points, we looked at our first objective, which was to describe these seasonal habitat utilizations. We saw that bathythermal niches differed among seasons, which was to be expected. We know that these fish are moving offshore in the summer and then moving back onshore during the spawning months. We also saw that individuals from the spawning population had annually consistent but different migration patterns. And so this is to say that all fish returned to Chameau Bay, but not all fish moved the same way while they were out in the lake. And so there were these different patterns when the fish moved out of Chameau Bay that we're able to see with acoustic telemetry. And notably, these were in Rochester and the Bay of Quinte, where these fish that migrated annually to either Rochester or Bay of Quinte stayed in these areas throughout the year, except for the spawning period. So this suggests to us that there was some type of feeding site fidelity here when the fish left Chameau Bay. The second objective, again, was to identify these spawning sites in Chameau Bay and then to look for spawning site fidelity. We found through acoustic telemetry and egg deposition that Cisco were spawning on shallow 30 meter rocky shoals. Kind of important to note here that the majority of our detections were only on 2 small sites. So these shoals within Chameau Bay were pretty small, and it seems like the majority of the production as far as egg deposition is coming off of these 2 areas. A little caveat here, we tagged and released the fish on these 2 sites as well. So if there is some type of site fidelity that's even finer scale than just returning to Chameau Bay, we would have biased our sample by tagging fish that were on those 2 locations when we collected them and tagged them. And so in the future, we may want to think about tagging fish from multiple areas within the bay instead of just on known spawning sites. We certainly see that with other species like lake trout as well. And then finally, 100% of our tagged fish returned in the spawning months, with the caveat that this is a very small sample size of 23 fish and that there were no females within our sample population as well. So some conclusions we can draw here, just to wrap it up. Given our data, we did not have any evidence for mixing between the Bay of Quinte stock with Chameau Bay during the spawning period. Certainly there was mixing outside of the spawning period, but not during the spawning, at least with males. The Chameau stock seems to be susceptible to different regional pressures because those fish are leaving Chameau Bay as a single spawning stock, but then moving throughout the lake as at different times of the year. They may be susceptible to these different regional pressures. For example, on the South Shore, we have a pretty large abundance of lake trout, Chinook salmon, which may have some overlap as far as predation. On the North Shore, there is still a small commercial fishery that these fish would be susceptible to that they're not on the South Shore. And finally, when we took a bit of a deeper look at the spawning shoals where these animals were depositing eggs, we saw quite a bit of degradation from things like silt, sand, and descended mussels. And so that's kind of led us down the path now, is how are these degraded habitats affecting early life survival? That kind of puts us into this next chapter where Brian Whittle has really been leading the way, looking at things like habitat remediation, habitat augmentation, habitat cleaning, and seeing how these might be a management lever that managers could pull, and further researching these so that we can provide that information to our resource management agencies. So that I'll just leave you with, there's a couple of publications that came out of the information I presented today. So if you want any more information, you can go look at those. One's about acoustic telemetry, the other's more habitat focused and gear oriented as well. With that, thank you very much. Appreciate everyone taking the time on their Friday to come out and listen to Kyle and I present about acoustic telemetry.
Well done. Thanks very much, Alex. Great talk. Just like Kyle's. Really nice to hear these complementary stories, at the most extreme ends of the basin, far western Lake Superior to eastern Lake Ontario. Interesting that both of you kind of saw some evidence for fidelity, staying within those bays for, at least, in your case, Alex, more spawning, but then they did the longer term sort of feeding migrations, sometimes halfway down the lake. Kyle, you definitely, one question I have for you, Kyle, to maybe to start it off with is, I don't know if you're limited by sort of that array that you showed us with the bathymetry depth and the difficulty of setting those receivers, but if you had sort of the kind of a broader coverage, do you think some of your western embayment ciscoes would have been taking some of those longer sort of foraging movements that Alex was able to see and you just weren't able to detect it?
Yeah, I think so. There's usually a period for a couple of months where the Cisco kind of go MIA and they're just outside of our array. And unfortunately, if you pull up the map on GLaDOS and you look at the coverage in Superior relative to Ontario, if they leave our array, it's our chances of them getting picked up go down quite dramatically, right? Unless they go to an existing project somewhere in a different corner of the lake. So, yeah, I'm sure that I think that we're probably seeing something similar to what Alex has seen. And I think that we'll probably get there in a couple more years once we have the data to help support that. But I suspect that, we'll see something similar to that. And hopefully there's some additional coverage in Superior going forward too, and we can kind of help actually visualize that, right? Instead of just saying that the fish go MIA for a few months, maybe we can kind of track them at least around the perimeter of the lake to some extent. That would be phenomenal.
Yeah. Okay. That's super helpful. So thanks for that. And then also, Jake Lace had a really nice question that others of you probably were going to ask. And he was able to, Kyle gave a really helpful answer to that in the chat if you haven't seen it. So let's move to now you can raise your hand in the chat. Like we've got Dimitri here ready to ask a question. You can also ask your questions in chat. But Dimitri, go for it.
Hey, great. Thanks. Awesome job, both of you. Great talks and great presentations. My question is for Kyle. I've been struggling with this issue with using predator tags in some of my other studies, especially because there's, we're looking at like stocking survival studies and we're doing predator tags, and we know that there's a large number of initial mortality, right? And then there's also on top of that, there's predation. Have you looked at like the fish's behavior before the predation event Because one of the things that I'm considering and I'm struggling you can't quantify it essentially, but like a fish could die from the stocking event or the tagging event and be dead on the bottom for a couple weeks before it gets predated up. And so I think, even though it's such a great tool to have these predation tags, I think we need to make sure that we do that. And I didn't know if you had seen any movement before predation events.
Yeah, so that's a great question, and thankfully again because we released our Cisco directly above a receiver, like there were some instances where a Cisco went straight to the bottom, was detected on that one receiver for 2 months, and then apparently it was predated. Right? So that did happen, but there were also instances where Cisco were swimming around Thunder Bay, kind of doing donuts for, just to put a picture in your mind, and then all of a sudden they got predated upon, right? So there definitely is an influence of fish that are probably dying as a result of the surgery and then being predated upon, but there is also more fish that are not dying as a result of the surgery that are being predated upon, if that makes sense.
Yeah,
It
yeah.
does exist for sure.
And then there's also the confounding part of like cold water slowing down the digestion of that predation trigger, right?
Yeah.
And so you again don't necessarily know how long until you know how long it is when that trigger goes off, but it could have been possibly in the stomach for a while.
For a while. Yeah. Yeah, it's definitely not perfect. But for our purposes, all we wanted to be able to know was like, okay, this is a Cisco, this is not a Cisco, right?
Yep.
And I think that we can get to that level at
Yep.
least. So for our purpose, it makes sense. But
Yeah, that's a really good point.
the
Yep.
points that you're bringing up are very valid, and they are things that we have considered.
Yeah. Oh, awesome. Thank you.
Yeah, thanks, Dimitri. Let's go to Tom, who has his hand up.
Unfortunately, my question is also for Kyle. But Kyle, sorry, Alex, but I'm just curious what your thoughts or estimates of the population sizes are between the three bays that you tagged fish in. Like, I'm just curious. You had the Black Bay fish that seemed to move a bit more as the population thought to be larger there. Like, I'm just trying to get an idea if that might play into how much spillover or movement we might see between those populations.
My guess is that Thunder Bay is likely the largest, followed by Southern Black Bay and then Nipigon Bay. But I think that what we might be seeing in Black Bay is that less of the habitat in Black Bay is ideal Cisco habitat, right? If you're just looking just at habitat, right? Thunder Bay would be the ideal Cisco habitat environment. So I think that similar to what Alex has seen, fish that might have some spawning site fidelity in Black Bay, I wouldn't want to be a Cisco living in Black Bay for a large part of the year. I'd probably go to Thunder Bay. It's deeper, it's colder, it's just better Cisco habitat. So we're probably seeing something similar to, fish having site fidelity in Black Bay, but going and feeding in Thunder Bay as an example, or elsewhere in the lake. Yeah,
Okay.
less walleye, less everything in Thunder Bay relative to Black Bay.
All right. So, and how does Nipigon Bay fit in there? Because it's even shallower, right?
It is shallower. That's a good one. Nipigon Bay is a little bit more of a wild card, I think. Definitely less Cisco there than in Thunder Bay for sure. Probably less than Black Bay, the southern part of Black Bay, but I'm not overly confident in saying that. And the fish in Nipigon Bay seem to move even less. Although I will say we only have 6 months of movement data from those fish in Nipigon Bay, whereas we had a year and a half from Black Bay and Thunder Bay. So this might, what I'm saying right now might change in a year's time. We might be saying, oh, the Nipigon Bay fish do move. It's just that we didn't capture that with the information that I presented here today. So you'll have to wait for a follow-up presentation, I guess, Tom, to see where the Nipigon Bay fish end up going.
Fair enough. Thanks, Kyle.
Thanks, Tom. So there's a question in the chat. Maybe we'll start with Alex because he's been thinking about more of the extirpation issues in Lake Ontario. And so John Bruner asked sort of how do you incorporate this idea of the high spawning site fidelity that you all are observing? How does that affect, I guess, a reintroduction effort from a management point of view?
Yeah, it's a really good question and something that we're trying to tackle right now. Part of the question is, when is that spawning site fidelity kind of imprinted on the fish, right? Is it something that happens like salmon homing, right? Is it some type of cue with smell or something like that? Or is it an adult behavior? Is it you're a schooling fish, you see a school of fish going somewhere, do you follow them there? So we don't have the answer for that right now. One of the things that we are playing with at USGS Tunison and Lake Ontario Biological Station is alternative ways of stocking fish. And one of those is experimenting with egg seeding. And so physically taking eggs from either, Chameau Bay population or from our research stock that we have on campus here, and then seeding areas with those eggs to see, one, do they survive? Can we even get them to survive? And then two, maybe down the line, because we've marked all these fish with genetics, do those fish return to those same areas as well? And so we're not there yet to answer those questions, but that's kind of where we're going with it. Thinking about alternative ways to put fish in the lake rather than just kind of stock and hope with juveniles.
Yeah, good answer. Kyle, did you want to add anything to that question? Okay. So let's see. There's another question coming in here from Etienne to Alex. After seeing high mortality of tagged females, how would you recommend trying to decrease tagging mortality in the future?
Yeah, I will try to answer this question with just some brief observations, and then I'm going to punt it right to Dimitri because he's the ones that actually tagged these fish for the study. When we tag female fish that we have here on station, so where I work, it's a hatchery that's been turned into a research facility. So we actually have fish here that we can experiment with and tag. When we tag female fish here, we tend to choose ones that don't seem as full of eggs. So maybe they've already dropped some eggs, or maybe they're just not as mature as some of the, or as fecund as some of the other females. And we decide to tag those fish because they have tighter muscles around their belly cavities, and it seems like there's more room to insert tags. I know in the past, Dimitri has experimented with actually physically removing some of the eggs from females before they were tagged, but I'll let him share his experience doing that.
Yeah, it's been a while since we've done that, but I'm trying to remember, the issue we had, of course, is you open that fish up and the eggs just come flowing out. They're so full of eggs. And whatever the buoyancy of those eggs are, and then you remove the space, whatever comes out when you open them up, and then you're putting the tag in, and that's displacing more eggs, and then you button them up. So whatever is happening with the buoyancy change with that, you're adding a brick and you're taking out what theoretically is maybe neutrally buoyant, or maybe it's fatty and it's a little buoyant. I'm not sure what how those eggs are when they're in the fish, but that is the main source of what we observed mortality from. The fish just could not maintain buoyancy. They could not maintain body position in the water. And, the ones that we removed a lot of eggs from and stuck a tag in, they did reasonably better. But then, who knows what happened to them out in the wild, because, the survival was terrible once we released them. And so it was just a very difficult situation. I don't really have a great answer for it, but it all is related to buoyancy and their ability to handle that.
Yeah, and I don't know if we have also to answer this question. I think Daryl was on the webinar before. He also was tagged Cisco, but I think also during the spawning time. And then I don't know if we have Todd Hayden on or not, but others feel free to chime in talking about sort of the best time to I mean, obviously spawning is nice because they're aggregated. They're potentially more susceptible to our gear. And then Kyle's idea of doing this in February is really interesting. If you can find safe ice, which you can't find in many of the lower lakes. So it is a puzzle. It is a challenge. So I don't know if anybody else that has tagged Cisco or is tagging Cisco, Nicole Watson's on the call, wants to sort of throw their camera on here on this topic before we jump to Bruce Morrison's question. Anybody else want to chime in that's in the audience?
Yeah, Bo, I can chime in really quick. So I have had similar issues when it comes to tagging Cisco during spawning. And basically what I was attempting is just trying to allow minimal number of eggs to escape from the incision. And conversations with Daryl just led to ensuring while I'm suturing to not trap any eggs within that incision itself, which can harbor infection. So combination of just trying to minimize egg loss, hopefully addressing the buoyancy issue. You're still adding the weight of the tag. And then we'll see. This is my first year tagging and the first time we've had the array set out, so we're not sure what our survival is. But yeah, it's a big challenge. And next year, we're considering using the Alouette trawl in the spring and potentially seeing about capturing the Cisco before they outmigrate into deeper waters and see if that changes, if we can catch enough fish at that time near Grand Portage, and to see how that helps with our survival. So that's a couple of things that we're going to be exploring on that issue.
Great. Thanks for chiming in, Nicole. A few comments, showing a comment there from Dimitri in the chat and also from Chuck Bronny in the chat, thinking about external tag options, which I think Mike Lowe is also pursuing sort of pilot work on that. So let's go to Bruce.
John Bruner's question about the site fidelity and extirpated fish at those sites that you're trying to stock. In your first, Alex, in your first map, I think you showed sites where ciscoes exist on the South Shore. And are those sites that were stocked? Or are they remnant populations like at Rochester?
Yeah, they were sites that were stocked, but we don't have any evidence that those fish, the larval fish that were collected there came from those stocked adults or juveniles, I guess. Yep.
No,
so they would have been
proof yet
wild.
that they spawned there.
That's right.
Okay.
We do continue with larval sampling and egg collections in the southern bayments, and every year we still collect larvae and eggs. So we're suspecting that there's still a population that's holding on down there, but we can't catch them with gill nets. We've tried that for years. Right now we've just switched over to using eDNA with Fish and Wildlife Service because that's the only way we can detect the adults in the southern embayments.
Thank you.
Thanks, Bruce. All right. Looks like we're slowing down on questions. Kyle, do we still have you?
Yep.
Okay. I guess I just wondered about what your expectation was regarding spawning site fidelity. I mean, Alex's study at least showed it really well for males. Did you expect to see that sort of spawning site or that sort of, I don't know if it's quite yet spawning site fidelity yet, but moving in that direction, the evidence so far that they really are remaining there, coming back to spawn in there. And that's not quite consistent with some of the genetic evidence that's been shown that looks like it's more of a mixed population in Lake Superior. So I wondered just your thoughts on sort of your findings so far and if that fit within your conceptual model or initial hypotheses.
That's a good question. So I think part of it too that's probably going to cause some issues if I really try to say that our fish have really strong site fidelity is the fact that we are grabbing them from a mixed stock, right? Like we aren't going to the spawning shoals during the spawning period, tagging them at the spawning location and then saying, look, they come back to the same spot, right? Like we're going after the spawn in Black Bay, Thunder Bay, Nipigon Bay, where they could just be a Cisco from all around the lake, theoretically, right? So that's probably going to be an issue that we face as long as we stick with the capture method that we currently have. We have had some internal discussions about trying to tag Cisco at different times of the years. So if that's a rabbit hole that we decide to go down to really try to nail down the site fidelity stuff, we'll probably switch up our tagging approach a little bit going forward. But it is interesting that we do seem to be having some early signs, I guess you could say, of some spawning site fidelity, although it's I definitely wouldn't be overly confident in stating that at this point in the study, based on especially based on the capture methods that we've been using. But yeah.
How long is your tag life? I mean, if you were to get repeated spawning, let's say in 2 or 3 years,
Yeah, so these V9 predation tags are good for 2.5 years. So we're a year and a half in already on our first batch of fish that we tagged.
So you might get 2 spawning times.
Yeah, that'd be
maybe not. Yeah, it'd be right on the edge maybe to get that.
Yeah.
Return. Okay.
Yeah.
Daryl, I saw that you sort of popped in there quickly. I don't know if you wanted to add something maybe related to the previous topic. Maybe we lost Daryl. Okay, well, I'm not seeing any additional questions from the chat. I don't think I missed any. Don Schreiner had one that Kyle answered there in terms of how many ended up going to the roe fishery. Maybe you all didn't see. Don't see anybody throwing their camera on to ask a question. Oh, look at that. Alina.
Sure. Hi. So I had something to build off that question about the roe fisheries. So Kyle, did you know that the commercial fishermen caught one fish? Is that because they had found it and they had reached out to you to let you know that they had a tag?
That was a funny one. Actually, no, I got a report that one of our fish was detected in Lake Erie at one of the ports. So some of the folks in Lake Erie at the Lake Erie Management Unit, I guess they go around and they just have a VR 100 and they just scan a lot of the fish that come to the dock apparently. And they just happened to get one of our Cisco that was at the processing plant down there. And then obviously that opened a whole can of worms because I was like, how did our fish get to Lake Erie without getting detected anywhere? And then eventually finally found out that it was captured in Thunder Bay and it just got on a plane or something on a truck and then ended up at Lake Erie and that's how it got detected. So yeah.
Okay.
It's kind of a fun one.
Is there other ways of determining if a Cisco has been caught by in commercial fisheries, or is it only a tag return or detection in a weird
It's only
way?
a tag returns. We're not externally tagging these fish at all. Whereas with a lot of our other fish, we're putting transmitters and we're externally tagging them, right? So theoretically, if one of these Cisco gets caught by commercial fishermen, if they're not chopping that fish up right there, they'd have no way of knowing. And they're working so fast, right? They're catching so many fish that even if there's a tiny little incision mark, they'll never pick that up. Yeah.
Okay, great question, Ilya. I thought you had buried the lead there, Kyle, with not telling us about a Cisco transmitting swimming all the way to Lake Erie.
Yeah, no, that was an interesting couple of days anyway, when we were trying to figure out what happened. That's for sure.
Okay, good. All right, well, let's see. Are we ready to advertise next week, Jake, or next month?
Yeah, so right now I'll post the registration link in the chat. So you should be able to, oh, all right. So if you follow that link, you should be able to register for the February talk. And then let me share the advertisement just for a sneak peek at that.
So we've got
Right.
webinars advertised or scheduled out until sort of mid-year, but we are always looking for more. So always very happy to have volunteers. Reach out to Jake or to me if you'd like to share your work, whether it's in progress or whether it's published, just to increase awareness of that. And so this will be a really great webinar next month by Trevor Pitcher. And hopefully many of you will be able to join. Amazing participation today. I never dreamed we'd get over 90 people on like January 3rd, with most people like on holiday. But thank many of you for joining in. It's an amazing turnout. It was just really, really cool. And a great job by Alex and Kyle in kicking this year off. So thanks everyone for joining, and we hope to see many of you back next month, February the 7th. All right. Happy New Year again.